Hello everyone, this is Kelly Vest, an undergraduate TA for Genes and Heredity, Fall 2015 semester. I'm making this video on complementation to try and clear up any confusion that might have occurred back on Friday, where you were taught a great deal of information in a very rushed and short manner. Therefore, I'm going to take some time now to go ahead and try and explain this in a way that might be able to help you understand this concept for your upcoming exam. Let's begin. Often, when we are studying genetics, we are looking at alleles. And these alleles are put into a shorthand, where capital letters code for dominant alleles, and lowercase letters code for recessive alleles. It would be a mistake to believe that these letters correspond to single base pairs, where there are two base pairs possible, where one codes for a dominant phenotype and one codes for a recessive phenotype, and therefore there are only two versions of these alleles. In fact, what we need to think of this as is a shorthand for a series of base pairs, where one combination, ATCCTAGGA, can code for a lowercase recessive allele of lowercase a. In fact, there could be hundreds of different ways that this particular sequence could code for a lowercase recessive a, and these would be mutations, denoted mutations 1 through however many exist. Mutations within a strand of DNA can change how it performs while still being distinct from other mutations that behave in the same way at the same locus. Let's look at a possibility. Given gene Scion7, which is fictitious, this gene codes for hair color in a normally dark species. If an individual is homozygous recessive at this particular gene, they will not express that dark hair color and will instead express white hair color. This is a non-functioning recessive allele, where the dominant A is the functioning allele. If an individual is A blank at this locus, they will be dark. If an individual is lowercase a, they will be white. There can be hundreds of mutations that can all equal lowercase a or capital A at this gene and produce the same phenotype as other equivalent mutations. Alternatively, there could be more than one gene within this phenotype that could produce the same white effect. For example, you could have Scion7 and GOSH4 both be involved in the pathway to white hair. Because of these possibilities, we do complementation testing. Complementation testing is where we take two individuals that exhibit one phenotype and we find out whether or not the mutations that they possess are within the same gene or if they are in two different genes. Let's look at mutations in the same gene first. We're going to be looking at Scion7, where two individuals having two different mutations each are homozygous recessive at this gene. Each of these mutations, therefore, is a recessive non-functioning allele, lowercase a. As you can see, each parent is lowercase a, and exhibiting the white phenotype. They each have two different mutations, however, they still code for that lowercase a. Because these mutations occur in the same gene and they have the same recessive genotype, all of their children are going to inherit lowercase a alleles and therefore exhibit that white phenotype. Though the mutations may differ, that recessive mutation still codes for a lowercase a and exists at the same gene. Therefore, the children are all mutant phenotypes like their parents. This is non-complementation. If there are two genes within the pathway to white hair, Scion7 and GOSH4, where one parent is recessive homozygous for one and the other parent is recessive homozygous at the other, where the father is capital A, lowercase b, and the mother is lowercase a, capital B. Both of these parents have a homozygous recessive allele at one of the genes within this pathway and therefore exhibit the mutant white phenotype. However, their children will inherit one capital A from the father and one lowercase b from the father, one lowercase a from the mother, and one capital B from the mother. The children are going to be heterozygous at each of these alleles. As we had previously discussed, being heterozygous dominant at this allele means that both of these genes will be functioning, and therefore the children express the normal dark phenotype. Because each child has one functioning allele at each gene, the gene pathway is functional. This is complementation, where one phenotype is governed by two genes, and individuals that are homozygous recessive mutants produce phenotypically normal children. So, if we have several individuals that have different mutations, we do complementation testing to find out whether or not they occur in the same gene, as mutations occur in all genes. So these mutations that are observed may be part of one gene or many. We use complementation testing to find out which mutations belong to the same gene. Given 7 white individuals from the same fictional creature, how many complementation groups exist, and which mutations belong to which group? We set up this table by listing each one of our mutations belonging to each one of these white individuals. We then set up another row of these same mutations, as we are going to cross each mutation with each other mutation to find out exactly how many complementation groups exist. Let's look at the completed table of crosses. Excuse me. As we can see, when 1 is crossed to 1, there is a minus sign. This minus sign means that the phenotype of the children is the mutant one. When we see a plus sign for 1 and 2, that means that the phenotype of the children is normal. As we saw back here, when non-complementation occurs and 2 mutations are for the same gene, the children are mutant. When complementation occurs and 2 mutations are at different genes, we see that the children are normal. Therefore, when we look at our map, we can see here that non-complementation has occurred. As we know that 1 and 1 will of course exist at the same gene as they are the same mutation, we know for a fact that non-complementation will occur, just as non-complementation occurs with 2 and 2, 3 and 3, and so on and so forth. We can see that complementation has occurred between 1 and 2, which means that 1 and 2 do not belong to the same gene. Let's go ahead and begin looking at this graph, and we'll find out exactly which genes belong to the same complementation group, which means that they belong to the same... Gosh darn it, where did I put that pen? It must have fallen behind me. They belong to the same gene. So we look across 1. We see that it complements 2, therefore they don't belong to the same group. It fails to complement 3, therefore 1 and 3 belong to the same group. 1 and 4 also fail to complement. That means that 1 and 4 also belong to the same group. And we can confirm this suspicion by looking at 3 and 4. If 1 fails to complement both of these and belongs to the same group as both, it therefore follows that 3 and 4 belong to the same complementation group. Let's look down at that cross. I look at 3, and I go across to 4, and I see that 3 and 4 also fail to complement. This confirms that 1, 3, and 4 are going to belong to group 1, or group A, as I have over there. Let's keep going. I look down at 7. I see that 7 complements 1, 2, 3, and 4. Therefore, 7 does not belong to any of these groups. However, 7 and 5 fail to complement. Therefore, I can put 7 and 5 into the same group. I can see that 5 and 7 both complement each other of these mutations, and therefore only 5 and 7 belong to that particular group. Therefore, I have now placed 5, 7, 4, 3, and 1. The only mutations that I have left to group are 6 and 2. I look down at all of 6's crosses, and I see that it complements 7, 5, 4, 3, 2, and 1. Because it complements each one of the other mutations, I know that 6 belongs to its own gene and its own complementation group. When I look at all of 2's crosses, it complements 1, 3, 4, 5, 6, and 7. Therefore, I know that 2 also belongs to a separate group. Try to do as much practice as possible with these kinds of maps, using examples from your book, from online, or even drawing up your own. By looking at this type of notation more and making your groups, you're going to find that it is going to become much easier, and soon you'll be able to do this very quickly. I hope that this video has been helpful in your studies, and good luck on Friday.